All right, so today we're going to go into protein synthesis. This process has two major steps. The first one is called transcription, and that's what we're going to be focusing on today in this PowerPoint. And the second is translation, which will be the next video. So this is where we really need to start focusing on the whole structure equals function thing. The order of our nucleic acids that we talked about last time is going to determine the order of our amino acids, which will determine each stage of protein structure, and then finally the entire protein folding. Now, you may very well be asked to describe what could happen if you break or change a structure or the process of some sort, and this is a really good place to start thinking about that. Before we can go into this whole process, we need to talk about and do a really quick review of proteins because it's been a hot minute since we talked about them and the structure of other various biomolecules. So protein structure, because structure equals function, so we aren't really going anywhere if we don't have the protein structure down. So basics here, the monomer is going to be an amino acid. Side note, remember that the monomer of a carbohydrate is a monosaccharide and the monomer of a nucleic acid is nucleotide. But here we're working with proteins, so our monomer is an amino acid. All amino acids have an amino group over here, a central carbon with like a little hydrogen hat, a carboxyl group, and an R group. Now, this R group becomes super important here in just a minute, and it's the thing that makes all amino acids different from each other. There's 20 different amino acids, and so there's 20 different R groups to make them different. These R groups might be polar, they might be nonpolar, they could be positive, they could be negative, acidic, basic, whatever. These characteristics help to determine the shape of the protein, which, remember, helps to determine that protein's function. Now, each protein is going to have at least 3 levels of structure, and then some of them, most to be honest, have a 4th structure. Each level is caused by a different type of interaction between the different parts of the protein. It's almost like structure of the individual parts of the protein helps determine its ultimate structure and function. Wild, I know. Now, this is what I want you to do. Go ahead, pause the video here if you're taking notes from it. I'm going to move on to the pictures here in just a second as I explain them, but if you want to take notes, you can do so. The primary structure here is the easiest to understand. It's just a chain of amino acids held together by the covalent peptide bonds. The secondary structure comes from the interactions between amino and carboxyl groups that aren't anywhere near each other. The peptide bonds leave partial charges on the groups, the amino and the carboxyl groups, which causes them to be attracted to each other. The two names for these structures are beta-pleated sheets and alpha helices. Tertiary structure is the final required structure. Remember how I said the R groups have different characteristics? Well, this is where that becomes important. If you have a hydrophilic R group, that whole amino acid is going to try its hardest to be on the outside of that protein. This causes a twisted kind of structure, and that's going to be our 3D major structure that we have here. Finally, you have the quaternary structure, which is where at least two tertiary proteins work together. You rarely have proteins that work alone, so you're going to have a lot of quaternary proteins. But if you were to break the tertiary structure in some way, then the quaternary structure wouldn't form at all. Proteins are ultimately super important. They're going to pop up everywhere, and they have about 1 million different functions. The 3 that College Board wants you to focus on are antibodies, enzymes, and quote-unquote structure. If you were paying attention this year, you probably remember that we talked about proteins a lot, especially in that structure function. All right, so now we can finally talk about protein synthesis. Pretty much we are just trying to get from DNA, which is stuck inside the nucleus, to RNA that can leave the nucleus. We're going to be working with individual genes here. One gene is going to give you one protein, except when it doesn't, which we'll talk about later. Proteins can only be made outside of the nucleus, either in the cytoplasm or on the rough ER. So RNA is ultimately super important. Big overview here. Transcription is where we're taking one gene of DNA and turning it into mRNA, which is then used in translation to make proteins. But we'll look at the the translation step here in just a little bit. Remember that eukaryotes, like us, plants, protists, fungi, other animals, just have to be complicated. Pretty much everything about protein synthesis in prokaryotes, like bacteria, is going to be much, much simpler. A lot of the things that have to happen in us and other eukaryotes just doesn't happen in prokaryotes. While we have to do things in small pieces and steps, prokaryotes, they just kind of say whatever and do it all at once. All right, so this is going to be another one of those pause the screen moments. While the structure of DNA and RNA is important, remember the theme of today is structure equals function. This is something we've gone over in great detail, both in Bio 1 and AP Bio this year. Besides, y'all don't just need me to read the screen to you. This is going to be an image of your basic DNA, RNA subgroups, so go ahead, take a look at this if you'd like. All right, so in Bio 1, we worked pretty extensively with these three types of RNA. And the good news is all of these forms of RNA still do exactly the same thing that they did when we talked about them then. So the three types of RNA we've already covered are mRNA, or messenger RNA, and that's going to carry the DNA message of the gene to the ribosome in the cytoplasm or on the rough ER. This is the one that we're going to focus most on today. tRNA, or transfer RNA, brings the correct amino acid to the ribosome based on the mRNA. This is used exclusively in translation, so more on that later. And last but not least, we have rRNA, or ribosomal RNA. Previously, we've just said that this is a structural part of the ribosome, which is absolutely true, but it also helps to connect the tRNA and the mRNA. But again, this gets into translation, so we'll come back to it. So remember how I cleverly mentioned that there are three RNA types on the last slide were ones that we'd already covered? Well, there's technically one more. snRNA, and keep in mind that the S and N are lowercase, just like the M, T, and R in the other types of mRNA, or in the other types of RNA. Sorry, my font's just weird. Is something that we call a spliceosome, which, as the name implies, slices things apart. We'll talk about that during mRNA editing in a little bit. Don't worry, I'm going to define the word intron here in just a bit. These are all of the pictures of RNA. Pause, take a look at them. You'll notice that each of them has a very different structure. This allows them to have different functions. And yes, I'm just as tired of saying that as you are of hearing it. The whole point of transcription is to take DNA that has to stay in the nucleus and make RNA, specifically mRNA, that can go out of the nucleus and into the ribosome. Now, one strand of DNA is going to have a bunch of different genes and is going to code for a bunch of different proteins. If each strand of DNA only coded for a single protein, then we would only be able to have 23 proteins, one for each coiled DNA chromosome. And given that proteins are functionally one of the most important macromolecules, that would be a very bad thing. There are 4 steps that have to happen in transcription, and they're all pretty self-explanatory, hence why this is slide 15 out of 21. You have initiation, which starts the process, elongation, which causes the protein strand to grow longer, termination, which stops the process, and mRNA processing, where RNA gets edited and changed around. Honestly, the most complex part of transcription is the initiation step, which makes sense because it's hard to get things started. You will notice, however, that initiation and elongation are going to be really similar to DNA replication. So there's a segment on the DNA strand called the promoter. That's pretty much the light up sign that tells RNA polymerase, the enzyme that makes this whole thing happen, where to bind. Remember in replication where we had about 100 enzymes that each did a specific thing? Well, here you have RNA polymerase, which is the only enzyme in the process that the DNA needs. It's just efficient that way. If you are asked to find the promoter on a DNA strand, you're going to look for something called the Tata box. I always remember that because I'm a 4-year-old and thinks that it's funny. Elongation is where RNA polymerase is going to add in new RNA bases to the ever-growing strand. Just like in replication, RNA bases can only be added to the 3' strand of the previous nucleotide. Elongation is much easier than replication, though, because you don't have the lagging strand that makes replication complicated. Next comes termination, which should really be the last step in my opinion, but alas, I didn't get to name things. This is where the DNA and RNA separate. Not much is known about this process, though. Apologies. Not much is known about this process, though, which is a good thing for the study for the AP exam point of view, but not so great for the, hey, we know a bunch about science thing. Now you just need to do the final edits to our mRNA. There are 3 different types of editing that we can do to our RNA. The first is adding a 5' cap. This is exactly what it sounds like. On the 5' end of the mRNA strand, you just add a piece of non-coding RNA. The point of this non-coding RNA is that it's going to protect the mRNA as it passes out of the nucleus and into the cytoplasm. It also tells the cell that it's ready to be translated. Next, we add the poly A tail. Pretty much here, you just stick a whole bunch of adenines to the 3' end of the mRNA. In the cytoplasm, you have enzymes that are always trying to break down mRNA strands. The point of this is to recycle RNA nucleotides that have already been translated. So it's ultimately good, but not terribly useful right now. The poly A tail allows some of the mRNA to be lost without any of the coding section going away. Finally, we have introns being removed from the mRNA. An intron is a segment of non-coding mRNA that needs to be removed before mRNA can be translated. The way I remember this is, quote-unquote, introns stay in the nucleus. The parts that are left, exons because they exit the nucleus, are then strung together so they form one complete segment of mRNA again. The thing that does the slicing and dicing is the fragments of snRNA mixed together with proteins called the spliceosome. Now, the weird thing about spliceosomes is that they don't always cut the same introns out every time. Depending on what introns are removed, the same gene can code for a few different proteins. That's why at the beginning of PowerPoint, I said that one gene codes for pretty much just one protein. And finally, after all that work in editing, you're left with a cohesive and mature piece of mRNA that's ready to go to the ribosome for translation. And that's where we're going to end today. More on protein synthesis when we come back for our translation video. Thanks, guys.